Hi everyone, thanks for coming. I'm really excited to introduce today's speaker, Dr. Michelle Afkhami, who's an associate professor at the University of Miami. Michelle did her bachelor's and master's degrees at Rice University in ecology and evolutionary biology, and then got her PhD in population biology from UC Davis. She then did her postdoctoral research at the University of Toronto and Michigan State before starting her lab at the University of Miami. She has a very productive lab and a CV that's really intimidating for a new assistant professor to look at, winning many awards for her scholarly excellence, receiving funding from the NSF, DOE, and USGS. She's also been recognized as a really excellent mentor and has advised over 100 research students and postdocs so far. Dr. Afkhami's research examines the interactions between plants and microorganisms, including important mutualists, as well as whole complex microbiomes from the genomics level all the way up to the community and landscape scale. Her research examines really fundamental concepts in ecology, like species distributions and biodiversity, as well as the hidden role that microbes play in environmental conservation and restoration and adaptation to global change. I think she's really at the forefront of plant microbiome research, moving far beyond just studying what microbes and how many microbes are in the soil and the plant microbiome, to ask important questions about their function and plant performance and plant community interactions and assembly. Before I hand it over to Dr. Afkhami, I wanted to remind you that we're having a seminar or a reception after the seminar up on the third floor kitchen, which I recently learned conflicts with an important grad student meeting. So hopefully some of you can still come. Anyway, please join me in welcoming Dr. Afkhami.
Okay. Hi, everyone. Thank you, Mia. That was so sweet. That was such a lovely introduction. And thank you guys for having me. So today I'm going to be talking about the ecology of microbiome-plant interactions, but trying to scale across multiple levels of biological organization. Okay, so in my lab group, we're really interested in species interactions. Excuse me, my voice is going from talking all day. All types of interactions, but a lot of the work that we do is between plants and their microbiomes, and we studied many different compartments of the plants and their microbial interactions. And of course, we're interested in negative interactions like pathogens, but we're also quite interested in mutualisms, where all partners benefit from the association. And of course, like most people who study species interactions, we're interested in the consequences of the interactions for the organisms that participate in them, whether that be the plants or the microbes. But what really gets me personally really excited is trying to understand how these effects scale across many different levels of biological organization. So sometimes we scale up and ask about the consequences for populations, communities, and ecosystems, and sometimes we scale down and ask about the genomic and molecular basis of these interactions. And so ultimately, what I'm hoping in the long term my research program is building towards is trying to really have a genes to ecosystem approach to understanding the importance of microbiomes in key ecological questions. But today I'm going to be talking about some of the work we've been doing scaling across several levels of organization, and hopefully in the future we're going to be scaling that whole distance. We've got a grant in right now to try to do that. Okay, so as I said, I'm going to talk about, in this case, 3 different areas of research that my lab is interested in, taking a multi-scale approach. So the first is multiple mutualist effects, or embracing some of the diversity within mutualistic communities. The second is how mutualisms and microbiomes can impact species distributions. And then finally, we're going to talk about the stress and anthropogenic disturbance, how that shapes the microbiome and plant-microbial functions. Okay, so I'm going to start with this multiple mutualist effects research. I will just say now that it's going to be a pretty bird's eye perspective. Like, I'm just going to highlight a few results from different papers. But then as we get into the later sections, I'll spend a little more time delving into a couple examples. Okay, so why I picked this picture for multiple mutualist effects is because for me, it's like, you know, I always think it shows so much complexity that it's really hard to study all those interactions. At the same time, it is like probably a big underrepresentation of the reality of the diversity that organisms interact with. So historically, mutualisms were studied pretty pairwise between like maybe a pollinator and a plant, or a guild like pollinators and a plant. But the reality is, whether we're talking about natural, urban, or agro-ecosystems, organisms are often interacting with many different mutualists simultaneously, or at least throughout their life. And so back when I was a grad student, I got really interested in the potential for conflict and synergy in multi-species mutualisms. And then more recently, my lab has continued to be interested in that and trying to understand that, to understand plant mutualisms and more complex microbiomes, and thinking about the underlying mechanisms. So why was I interested in this topic? Well, that's because there are some things that are unique about multi-species mutualism that you really don't get when you study pairwise interactions, or like pairwise mutualistic interactions, or even multiple negative interactions like multiple predator effects. So to illustrate that, I want to use an example, which is the study system that I'm going to be talking about for a lot of this section. So I'll go ahead and introduce that. So that's the association between legumes and rhizobia and mycorrhizal fungi. So I love this system because I feel like it's a good both genomic model system and ecological model system. So for instance, in this case, we have genomes for all the participating species, and for the plant, we have HapMap projects, we have mutants, and so on. And it's, I think, a good ecological model system because it's really like the most straightforward, simplest multi-species mutation you can have. Like, there's only a few species, but we know that it's both ubiquitous in nature and ecologically important. So for instance, this in a paper, actually in collaboration with your own Marge Weber, we established that a large portion of legumes, maybe it's not exactly 90%, but a large portion of legumes, which is the third largest plant family in the world, associate with nitrogen-fixing bacteria that, of course, provide nitrogen to the plant in exchange for photosynthetic carbon. And then also, others have established that something like 80% of land plants associate with mycorrhizal fungi, which can provide resources like phosphorus uptake. Okay, and of course, it's really important for things like succession, crop rotations, and so on. Okay, so this all started with like a simple thought experiment. So of course, historically, we're thinking about mutualisms as pairwise. If it's a mutualism, we should see an increase in performance of a host organism if it grows with a partner that is a mutualist, at least some of the time. But the real question was like, well, what happens when you have multiple partners? So you could see an additive effect where the whole is essentially just the sum of the parts. But we might actually expect something like synergism, where the whole is greater than the sum of the parts. And that could occur if there is complementarity of the rewards being conferred by the different partners. So for instance, in the case of this plant, if it's co-limited by both nitrogen and phosphorus, having partners that provide primarily different rewards could increase performance above and beyond that expectation by removing co-limitation. And so essentially this is something that's pretty unique to multi-species mutualisms because it requires complementary benefits from multiple partners. In contrast, though, you can also see the opposite effect, where we have, when you have multiple partners, you actually get less benefit than having a single partner, or possibly even having no partners at all. And so there's really this big, well, okay, so why would that happen? So for instance, if you think about, like in the case of this plant, right, the rhizobia, the mycorrhizal fungi, and pollinators, ant defenders, and so on, all receive photosynthetic carbon as a primary reward. If that's a limited resource, which it may well be, they could compete for it in a way that could have cascading negative effects on their host, sort of a tragedy of the commons type scenario. And so I was really interested in, like, you know, what are the mechanisms that allow organisms, well, first of all, like, when do we see these different types of outcomes, and what are the ecological mechanisms that can explain them? So why do we sometimes see synergism and sometimes see additive effects and sometimes see antagonism? And so initially, when I first got interested in this, I was thinking a lot about the ecological mechanisms. So things like the overlap and the rewards being conferred by the partners, so like the example I gave you with complementarity, the spatiotemporal reliability of partners, the cost of the benefits being provided, all these kinds of things could play into it. And there's been a lot of beautiful research by a whole bunch of groups over the years testing a lot of these hypotheses. But in the meantime, I got really interested in thinking about the molecular mechanisms that could underpin how organisms can maximize the benefits and minimize the cost of associating with multiple partners. So as a first step, we decided to do a study, a transcriptomic study, where I grew plants in the presence of no partners, just mycorrhizal fungi, just rhizobia, or both partners together, and then did, of course, looked at the things like performance of the plants, but then also looked at the gene expression profiles across the genome to look at how having multiple mutualists would change the sort of molecular phenotype. And the first thing I want to show you, there were many results in this paper, I'm just going to highlight a couple, which was this PCA, which the two axes explain something like 60% of the variation in gene expression. And it turned out that there was really, each point represents the transcriptome profile of an individual plant, and each color represents a different microbial environment. And you can see there's really strong clustering by microbial environment, indicating that these multiple mutualists were having sort of pervasive genome-wide effects on expression. And when we looked at how well this variation in expression could predict plant performance in our experiment, it explains something like, I don't know, like 30% of the variation in plant performance. So we were able to see that the multiple mutualists were having impacts on gene expression that were tied to the changes in fitness that were non-additive. We also identified some candidate genes for important sort of regulatory points in networks. This was from a differential expression analysis where we found groups of genes like this one where we called them reversals, where when you have a single partner, you see upregulation and expression of those genes, and then when you have multiple partners, you actually get the exact opposite, a switch point, where you start to see downregulation. And so we think that these genes could be very important in how organisms regulate multiple mutualistic interactions. And we did later build co-expression networks and found that these genes are really, really highly connected within the network, suggesting that they're potentially very important for coordinating information among genes. Which leads me to the second study, which was actually led by an undergrad in the lab at the time, Satvik Palakirti. He's now an MD PhD student at WashU, but we still keep in contact. And this study was really one of those things where it came from my realization that I needed to learn more about molecular biology. So as an ecologist, I still need to learn a lot more about molecular biology. But as an ecologist, it was like super obvious to me that organisms don't occur in a vacuum, right? We need to embrace the complexity of organism-organism interaction. So I needed to add, consider the idea that multiple mutualists matter. But then after doing the first study, I was like, oh yeah, like genes, they don't occur in a vacuum either. And so if I really want to understand the consequences of these complex interactions, I need to move beyond a gene at a time approach and look at, co-regulation across the genome. And so that was what the goal of this second study was. And we did co-expression network analysis, where we looked for gene modules or groups of genes whose expression changes in tandem, and evaluated how the mutualist environment changed or rewired those modules. And so you can see here, I'm showing you an example of just one module. This is with, sorry guys. This is with no partners, just rhizobia, just mycorrhizal fungi, or both. And it's the exact same module, but I'm showing it to you as a heat map in those different environments. And in the heat map, each pixel is a representation of the co-expression. It's the correlation in the expression between two genes. And so, you test this statistically, but you can tell visually that when you see really different patterning, that's an indication that there's really big changes in the co-expression network structure. And so you can tell in this case, like in the presence of multiple mutualists, we're getting a really different co-expression network structure. And it turns out many of the gene modules were non-additively affected by multiple mutualists. And in fact, of the gene modules whose expression profiles could explain variation in plant performance, 80% of them were non-additively affected by multiple mutualists. So what that indicates to me is that all right, the mutualists are having non-additive effects not only on individual genes, but also on the relationship among genes in ways that can be tied to the non-additive effects on plant performance. And so there's still many more steps to really get at the mechanism underlying synergism and antagonism, but there's indications that we're making progress towards that. Okay. So then we were interested in thinking about the evolutionary consequences because a lot of the multiple mutualist literature is coming from a very ecological perspective. And of course, we were interested in how does that affect the evolutionary trajectories of the participating species. And so in this study, we took 200 genotypes that have been previously sequenced of this Medicago plant and grew them in the four different microbial environments, and then did genotypic selection analysis. And what was interesting, some of the key results were that the presence of multiple mutualists had synergistic effects on selection on plant traits. So we saw like double the strength of selection on some of the key plant traits associated with architecture of the plant. In addition, we also saw that when we had multiple mutualists, there was actually greater fitness alignment between the symbiote and the plant, indicating that, that potentially strengthening, by having that third party, in this case the mycorrhizal fungi, we saw actually a strengthening of the relationship between the rhizobia and the plant. And so overall, our takeaways from this was that, there seems to be likely microevolutionary trajectory effects of the multiple mutualists on the participating species. Okay, so I told you, bird's eye view. So what I like about the system is, of course, we have the ability to ask questions at these lower levels and ask about, all the way up to asking about simple community dynamics. As we move forward, we're trying to expand out a bit across a few more levels of organization. So for instance, we're trying to embrace a little more complexity within the communities. So for instance, this is a project my graduate student, Amanda Rostern, has been integrating pathogens into this framework. So she did an experiment where she manipulated the presence or absence of these two partner mutualists, but then crossed that with the presence or absence of a pathogen. And so she's been, she actually is waiting for her data to come back from TAG-seq so we can look at the sort of regulatory responses. But she also looked at the fitness consequences, and what was really fascinating from this work, we haven't even finished the manuscript yet, but she found that genotypes of the plant that are resistant to pathogens are also better able to, or more often get synergistic effects in multiple mutualists and have much fewer antagonistic responses to multiple mutualists. And likewise, on the converse, the genotypes that are susceptible to pathogens see a lot more antagonistic effects of multiple mutualists compared to synergism, suggesting that there's something about how plants regulate their pathogens that also help them maximize benefits in complex mutualistic interactions. Okay, we also are trying to expand down to ask about complex ecology at the genetic level. And so Damian Hernandez, who is a former grad student, he's now a postdoc at Toronto and Amsterdam, but he did this project, and the paper is available on preprint, still kind of in the review process, but he was interested in how do we regulate context dependency and match the environment and species interactions. And so he actually identified expanded gene families as a genomic mechanism that allows that molecular flexibility. And so he did a comparative genomic analysis comparing plants that associate with mycorrhizal fungi to plants that don't. And he was able to identify 430 gene families that are expanded in mycorrhizal lineages. And those gene families seem to be really important for context-dependent regulation of the mutualistic symbiosis under different stress conditions. So for instance, he found there was about a 200% greater context-dependent expression within these expanded gene families under stress and in association with mycorrhizal fungi. There are also hotspots for SNP variation associated with fitness benefits from the mycorrhizal fungi. And those expanded gene families are, so that was from transcriptomics and genome-wide association mapping, to be clear. And then also from gene duplication analysis, he found that they're very highly enriched for tandem duplications, suggesting that there's sort of this continuous supply of genetic variation coming in. So I'm super excited about this stuff, but I don't have time to talk about it in detail. All right. Okay, so we're in the second section. So very similarly, I guess my point is like, obviously, we use multiple scales to get inference about that multiple mutualistic interaction. And likewise, we've been applying a similar perspective to understanding species distributions, although we're looking at different levels of biological organization. And my interest in species distributions very similarly started as a graduate student. I was, as a graduate student, sort of like the major contribution of my dissertation was to show that associating with mutualisms can expand species niches, leading to range-wide expansion over large geographic scales. And so that was done in California with a fungal endophyte that, and grass association, showing that essentially the fungal endophyte allowed them to get into drier parts of the species range. And so I really liked that work, and I thought it was meaningful, but, oh, and I will say I've been back. I did a sabbatical project, and it turns out that 15 years later, having endophytes historically means you're way more likely to persist. So populations were half as likely to be locally extinct if they had mutualisms. Probably, it looks like probably due to climate change differences, but anyway. So that's been really fun, but I guess the issue is that this is an exclusively vertically transmitted symbiosis, tightly co-evolved, so there's a high coupling of the reproductive success of the host and the symbiote. And so but the reality, and so that's very important, and symbiosis is very important, but the reality is that there's also we're like living in a microbial landscape where organisms are having interactions with like many diffuse interactions with microbes. And I wanted to know to what extent these sort of microbial landscapes that are not necessarily tightly co-evolved might impact species distributions. And so that's where we've been working now. Okay. So before I go on, I guess, why should we care about the factors that determine species distributions? I'd argue it's a conceptually interesting question in its own right, but I think it's also very important for understanding ecological things we care about. So for instance, quantifying invasion potential of exotic species, managing and restoring endangered species, which some of our work has been contributing to. And forecasting biodiversity's response to anthropogenic global change. So, I think it's valuable. It's, fundamental ecology, but also has applied value. Okay, so we've been addressing this in the Florida scrub ecosystem. So the Florida scrub occurs along in this green area called the Lake Wales Ridge. Historically, long ago, when Florida was underwater, that was an island, and so it has the highest rate of endemism in the southeastern United States. And so it also has many species that are imperiled or endangered because there's only about 15% of its historic distribution remaining. We work a lot at Archibald Biological Station, which is here at the southern tip, but we've more recently been expanding out to a lot of other sites. This is what the Florida rosemary scrub looks like. It's characterized by this dominant shrub right here, the Florida rosemary, and these large open sand gaps that host a diverse assemblage of herbaceous species, many of which are endangered and endemic. And the reason I was super excited to study this system when I moved to Florida was because Archibald has this beautiful history of studying plants at this here. And so they have long-term demographic data for 20, 30 years, tracking many individuals. They know a lot about what shapes plant communities and so on. And so it opened up a lot of potential to ask questions about plant-microbe dynamics that might be challenging in other circumstances. So some examples of things that we know are super important to plants here. Elevation matters a lot. This habitat is considered, I know, high elevation. It's not, but I mean, it matters. So Florida's really flat, guys. But the thing is a handful of meters higher, but it matters a lot. Look, I mean, we get really big turnovers of plant community, and the reason it matters is because it's like a greater distance to the water table, so this habitat is a lot drier, so effectively a lot more stressful. Also, we've shown in our own analyses that nutrients are much poorer in this habitat than the slightly lower elevation sites nearby. So elevation relative to the water table matters a lot, and that's how we measure it, is relative to the water table. Fire, pyrogenic ecosystem. Fire comes through, creates large open sand gaps that are gradually recolonized by the Florida rosemary and that herbaceous assemblage. And then if you don't have a normal fire return interval, you get woody encroachment, and you lose those gaps, which host a lot of those endangered and endemic plants. So management with fire is very important in this ecosystem. Okay, so we've worked on a whole bunch of species now in the system. This was the initial suite of species that we started with. They include a lot of different life histories. They all occur in those open sand gaps, and they include some of the endangered federally and state listed species. All right, so this part we're sort of scaling up. So we're starting at asking about the consequences for microbes at the level of the individual plant's performance, how that in turn can scale up to impact population dynamics, and then how that in turn could impact species distributions. So the thought process here was that, okay, we need to understand the consequences for like individual level fitness effects, but then if we want to understand species distributions in a sort of ecologically mechanistic perspective, then understanding why some populations flourish and other populations decline could be really useful, since of course species distributions are comprised of many populations across the landscape. And so that was the initial thought process. I'm going to show you the sort of initial studies we did and then tell you about what we've been doing since. Okay, before I go into this first question, I want to acknowledge two of my long-term collaborators on this, Chris Searcy, who's another faculty member in our department, and we co-advised Aaron David as a postdoc in our labs, and now he is the director of the plant lab at Archibald Biological Station, so he runs his own group and we collaborate quite often. Okay, so the general approach was really straightforward at the beginning, right? We collected microbiomes across the landscape that varied in important environmental gradients like time since fire and elevation. We knew that this was made possible because Archibald has a 5 meter by 5 meter grid across the landscape, so you can get all of that data. And then we did the very straightforward manipulation of the microbes and grew our 12 plus species in each of those different environments, so that essentially we're looking at how the presence or absence of the microbiomes interacts with these environmental gradients to affect plant performance, just for that individual level stuff. All right, so I'm just showing you one life history stage. This is early growth, and I'm showing it to you for the 12 different species over here. Each pair shows you with microbes, without microbes, and 6 of the species responded positively to microbes, 4 negatively, and 2 didn't respond. Okay, so it turns out when we look across different life history stages, like every plant species we've examined responds to the microbiome at least some of the time. My other main thought when I first saw this figure, which was like one of the first figures we generated in the system, I was like, okay, lots of significant effects, but is that magnitude biologically significant? And so what it turns out is that this is the average effect across all microbiomes, but environment matters. And so when we broke it down and looked at the interaction between the microbial community and environmental gradients, we saw that the magnitudes of the effects can be much larger. So this is an example. This is an endangered St. John's wort. And I'm showing you 2 vital rates, germination and early growth. And I'm showing you them across an elevation relative to the water table gradient. And you can see, that the microbial effect on the positive microbial effect on germination, like increased in strength with increasing distance to the water table, so in those more stressful habitats. And interestingly, we also saw a strengthening of the negative effects on early growth with increasing distance to the water table. And we saw this sort of thing for many different species where environments by microbes showed a strong interaction. Also, really importantly, is that as I kind of pointed out already, like different microbes are having different effects at different life history stages. So how do we know how the microbes are actually impacting the plants? We really need an approach that allows us to look across multiple life history stages. And so that was something that was possible when we started doing the population work, because we were using demographic modeling, specifically integral projection modeling. So the idea here is that you model population growth as a function of survival, growth, and fecundity, and it incorporates many different life history stages. So essentially, we built those models using long-term demographic data from field surveys across the entire landscape for the plants, and then we were able to adjust them based on our experiments to look at how the microbial effect on these different vital rates might impact population growth rate across the landscape. And so that's what we did. I can tell you all about it, but I'm just going to go during question and answer if you want, but I'm going to go straight to the result for sake of time. All right, so this graph is showing population growth rate on the y-axis, so lambda, and it's actually the mean across 92 different patches. This is a naturally very patchy habitat. When lambda is greater than 1, the population should be increasing. When it's equal to 1, the population is stable. And when it's less than 1, the population is decreasing. And I'm showing it to you across a time since fire gradient, since that's a really important environmental feature or factor in this ecosystem. And so when we parameterized the model based on the field data and then adjusted for our experimental plants that had the natural microbiome across the same gradient of environmental conditions, we saw it looked very, very much like what happened in the field. There's a period of positive population growth rate from like 3.5 to 7.5 years post-fire, which is very, very important. It's been shown in other work, very, very important for population persistence of this. This is actually, that's endangered St. John's wort for this species. What was interesting is then when we adjust the model for the hypothetical world where you could remove endophytes from a landscape, we really almost never saw the populations reaching that threshold of lambda equals 1. And so it seems that if we did not have these sort of diffuse interactions with the soil microbiome, then this endangered species may not really persist on this landscape. We wanted to investigate this a little bit more, so we looked across the maximum population growth rate across the different sites in the landscape. So here I'm showing you each point is the max population growth rate for one of those 92 populations. And you can see they vary in where they are in terms of elevation relative to the water table. And so I think one thing that's important to point out is, in fact, that high elevation sites are in fact really stressful for plants, because you see that they actually do have decreased population growth rates in those conditions. And we see it in the field too. But I thought what was really interesting is that you actually get a bigger divergence between this cloud of points and this cloud of when you're in the higher stress conditions. So at high elevation, the positive effect of microbes on the growth rate of the population is actually stronger, suggesting that the microbiome is most important for population growth rates in high stress environments. So again, there seems to be evidence for this interaction between environmental variation and the microbiome for impacting which, in this case, which populations do well and so on. Okay, so that made us think, of course, that maybe this will impact the landscape scale species distributions. This is something we're doing a lot more rigorously at this point, but I'm going to show you actually the very first paper we published on, and then I'll tell you a little bit what we're doing now. So this was done by Kum Thapamagara, who was a postdoc in the lab at the time. He's now actually he's a postdoc in Colorado, so I'm very happy for him. And he did a study where he, similarly manipulated the microbiome, accounting, I should say we always account for all the sort of issues with nutrient deposition from autoclaving and so on. And crossing that with benign versus allelopathic stress from that dominant shrub, the Florida rosemary. And the idea here was that he was curious about how the microbial effects in allelopathic conditions compare to the microbial effects in benign conditions. And we were curious whether this difference, which we use as an index, would be predictive of how likely these different species were to occur in a landscape where allelopathy is really common. And that was made possible by comparing the results from Coombs' study to a paper that had been published by Eric Mingus. So he was the former director of the plant lab at Archibald, and he had published a study that had looked at this, the whole community in these open sand gaps and their frequency across the landscape, across many, many different sites. And there were a number of results, but I think sort of the nutshell result is that the degree of microbial mitigation or exacerbation of allelopathic stress was like very highly predictive of patch occupancy across the landscape. So, okay, what do I mean? Essentially, we calculate this index for which determining whether microbes mitigated that allelopathic stress for the plant or exacerbated that allelopathic stress. Sorry, guys. And then we correlated that with the patch occupancy on the landscape. And even though we only had a handful of species, it actually explains something like 80% of the variation. So it seemed very highly predictive. And it turns out, I will just say that we've done this for a lot more species and it still falls out, very similar pattern. But the point is that species like this one, this is a Lechiodacurtii, where microbes mitigated allelopathic stress, they were a lot more common on the landscape where allelopathy is frequent than species like this Liatris tenuifolia, where the microbes actually exacerbated the stress. And so again, sort of the interaction between microbes and the environment impacting where plants occur on the landscape. So the general approach here has been like sort of individuals to populations to communities. We've been doing this a lot more rigorously recently. We have an NSF grant on microbial landscapes looking at how environments, we're looking at how microbes impact individuals, then populations, metapopulations, and distributions. For at the level of individuals, we've been looking at how different features of the landscape shape the microbiome in a way that has cascading effects on hosts. So looking at diversity and composition and all those things, but also more recently, we've also been looking at the functional changes in the microbiome in terms of the metatranscriptomic responses. And this was done by former postdoc, Dan Revellini. He's now doing a Marie Curie in Spain, but he and I still collaborate all the time because we're still writing more papers. And so that's been quite interesting. But then I think what I'm really excited about right now is these middle steps, which is being done with a postdoc, Josh Fowler. And we're creating more demographic models using that long-term data in our experiments. But what's neat is that one, we'll be able to look at which features of the plant life history are most important for predicting when microbes matter. And we're incorporating metagenomic data, so we can look at how variation in functional attributes of the microbiome might impact the importance of the microbes for population growth or decline. So I'm excited about that part. And then we've done a lot of the work on the other bookend. In this study, we're writing it up right now, but we've been out to 700 sites in the landscape where we collected the microbiome and sequenced them, and then also surveyed all the plant species. And we have the grid data on the abiotic data. And so we've used the abiotic data and the microbial community features with species distribution models to predict the distribution of all the different plants, to see what features of the microbiome consistently predict plant distributions. And so it's not published yet, but some of the key features seem to be bacterial diversity, fungal pathogens, not that surprising, and genes involved in phosphorus uptake seem to be very important in this ecosystem. And mosses do something different than a lot of the other plants. Okay, so as you can see, this is another approach where we've scaled across multiple levels of organization. And that has also been very useful when we've been thinking about stress and anthropogenic disturbance. And this area of research really is something that's been an outgrowth of starting my lab in Miami, where we have these really unique ecosystems, but they are all highly imperiled because we have these large human population centers, and so none of the ecosystems are unaffected. So as a result, we've been studying a lot of different aspects of anthropogenic stress. We've looked at fire, also salinity stress from sea level rise. So we've been working on mangroves and how the endophytes with a stress legacy experience with salt stress are better able to confer resistance to salt as we get, with sea level rise, we're getting increases in salinity in our aquifers. And then we've been doing some more applied research, actually working with the Water Management District to look at microbiomes and tree islands to help understand reconstructive restoration and how we can use microbiomes to assist in that. And then the last thing I'm going to talk about is, the one I'm actually going to talk about is fragmentation. So habitat fragmentation, I should say that work was initiated by Casey Kieswater, a former grad student who's now postdoc in New Zealand. And she really got this work going and the system going, and that we're pursuing more stuff now that I've got some new students working on it. All right, so of course the idea here is that, in a lot of cases, if we didn't have any sort of human disruption, you might have a continuous native habitat across a landscape. But then if you have, human impacts, you can get the introduction of a novel matrix habitat into the landscape, which essentially leads to the breaking up of the natural landscape. So this has myriad consequences for how organisms experience their environment. So for instance, for one, you have now a novel habitat with different stressors now on the landscape. And then you also have greater habitat isolation between native patches. So because of all these and more impacts of habitat fragmentation, it's not surprising that scientists have rated habitat fragmentation as one of the biggest threats to biodiversity, both in their own systems and globally. And as a result of that, it's not that surprising that people are really interested in the consequences for predominantly plants and animals. And so I think this is really important. But there are some taxa for which we have very, very little understanding of how fragmentation impacts them. And so, of course, I've already highlighted this study where they were like, this was 2017, but it's not that, we haven't gotten that much further, where microbes are just, we don't have very good understanding. And this is, of course, concerning because we know that microbiomes can be really important in ecosystem functions like carbon sequestration, nutrient cycling, decomposition, and so on. I think what really surprised Casey and I, though, even more so, was that we couldn't find any studies that experimentally tested how the effect of fragmentations on the microbiome might cascade to impact the plants. And why do we think that was surprising? Well, one, obviously, people are really interested in how fragmentation impacts plants. And so there's a lot of emphasis on that. And then two, outside of the fragmentation literature, of course, we've already talked about, we know that microbes can have big impacts on plant health. And so this was sort of the, one of the key goals of her dissertation work was to try to understand the impact of fragmentation on the microbes and how that could cascade to impact plants and plant communities. So, the questions that I'm going to talk about today focus on 3 questions from 2 of her papers. So the first is, does anthropogenic habitat fragmentation affect the soil microbiome? Does that in turn impact the plant performance? And then could those effects cascade up to impact community dynamics, right? First part I'll talk about is from one paper. It addresses those first 2 questions. Before I do that, I better introduce the system. That's the pine rocklands. It is a really local system. So it occurs really only in South Florida and a little bit in the Bahamas. That's it. And it's characterized by this open canopy of slash pines, this sub-canopy of palmettos, and then this understory of herbaceous plants. And unfortunately, it is really highly imperiled. There's only about 2% of its historic distribution remaining outside of, there's this one bigger patch in Everglades National Park. But this patch has really different geology and different ecology. So so this gray is what was the historic distribution and the pink is what's left. That's because it occurs on what's known as the Miami Rock Ridge. So, which is, again, slightly higher elevation, but again, matters a lot. So that slight higher elevation means that when you have storm surge, your house is less likely to get flooded, or your business. And so people have disproportionately built on the Miami Rock Ridge because that small difference in elevation makes a big difference in case of like a hurricane. And so unfortunately, a lot of the habitat has been destroyed, leaving it very fragmented. But I will say that the Miami voters actually raised a tax, the Miami-Dade raised a tax specifically to buy up what's left of it. So almost all of it is protected at this point. Okay. So Casey has been to many of these fragments. She's been to probably 30 or 40 of them. We've surveyed them for various projects. In this case, for this first one I'm going to talk about, she went to 14 different fragments across the historic distribution of the pine rocklands on the Miami Rock Ridge. And at each site, she collected soil microbiomes for sequencing and for use as an inoculum in an experiment. And at each site, she characterized a bunch of different habitat features like the soil pH, fire history, and things like that, but also used a few different metrics to characterize the degree of habitat fragmentation. And then she brought them back to the lab, sequenced the microbiome. At this point, we were doing more amplicon sequencing. We're really, I think almost all the projects in the lab are now moving to metagenomics, but it was cost prohibitive a few years ago. So she did 16S and ITS, I think. And then, but what was cool is that she could still get at some of the functional consequences by looking at her experiment where she inoculated with all 14 microbiomes and sterile controls to look at the effect on 3 of the different native plants. Okay, so the first main result, I know, kind of a mess, but let me just highlight a couple. So the first thing is that the legacy of fragmentation appears to have a big impact on the soil microbiome. So for instance, I'm showing 3 out of the 4 primary axes of variation in community composition on the y-axis of these. So 1 for fungi and 2 for bacteria, and they showed a significant relationship with different metrics for habitat fragmentation. So it looks like 3 out of the 4 primary axes of variation in community composition responded to fragmentation. We found diversity also responds, functional gills and so on. Then the question was, well, does that actually matter for the plants? And so what we did is that experiment, and then we looked at how the variation in the microbial community affected the different aspects of plant performance. And so we found that all 3 of the axes of variation in the community composition of the microbiome that were affected by fragmentation legacy were also able to explain variation in plant performance. Okay, example. So here I'm talking about this fungal axis. So performance of the plants generally increased when you had high values of this fungal PCO2, so it's a principal accordant axis. Anyway, and microbiomes that have high values of that are microbiomes that come from areas with recent fires and high connectivity. So that means low fire suppression and low fragmentation. So these are fragments that are much less impacted by fragmentation than other sites. We also used random forest models to identify microbial families that were most important for predicting plant performance in our experiment. And about 1/3 of those were, you could call it fragmentation responding. Their abundances depended on the degree of fragmentation. And so that gives us some candidates for which microbial taxa might be driving the fitness differences and tying that to that effect of fragmentation. So we've got a set that we can follow up on the future. Then everyone was curious about whether this in turn impacted community dynamics. And so Casey did a follow-up study with an undergrad in the lab, Ladiana. And in this study is very similar in some ways. Like she went to 15 different native fragments, but in this case, collected microbiomes both from the core of the habitat patch and the adjacent matrix. And then she characterized a bunch of the environmental features, the degree of fragmentation, all that stuff. And then characterized the microbiome again from the 30 different microbiomes, and then used all 30 microbiomes plus all the sterile controls in an experiment with a mesocosm of understory plants from the pine rocklands. And let's see. So the first thing we learned is urbanization has massive impacts on microbial communities, at least in our system. Like, it is the biggest effects we've seen just consistently across all metrics of composition, diversity, whatever. So here I'm showing you diversity effects. So you can see if you look at the urban matrix, that we saw a decrease in diversity for the bacteria. That was due to there's about 200% higher richness in the native habitat for bacteria. So we broke it down into richness and evenness to figure out how diversity responded. And evenness was unaffected. But in the other case of the fungi, we actually saw the opposite, where the urban environment had higher diversity, which we thought was weird. But it turns out that richness is equal, but it's way, way more even in the urban environment. So we think that it may be just more homogeneous. And so in that case, the evenness won out since richness wasn't very much affected. Composition also responded. What was really interesting was for the taxa that we could map to potential or putative functional groups, we saw that there was a big increase in the symbiotrophs, or potentially symbiotic beneficial microbes for plants, and a big decrease or a big increase in the urban environments of pathotrophs or pathogens. And so there was this indication that likely we were having a shift from a plant beneficial microbiome in the native habitat to an antagonistic one in the urban environment. We then looked, of course, well, did it actually impact plant productivity? And so we first looked at the native microbiome, and you can see that the native microbiome, when plants were growing the native microbiome, they did way better. I think the productivity was almost 300% increase. And so that suggests that the productivity of the native plants on the habitat fragments depend probably pretty heavily on a healthy native microbiome. But interestingly, for the urban communities, that just wasn't true. So we didn't see a significant increase in performance with the microbes, but the mesocosms actually had pretty high productivity. And it turns out that there was a 60% increase in nitrogen and a 500% increase in phosphorus in the urban environment. And even though we added just a tiny bit of that soil to our mesocosms, there was just way more nutrients. So I think what that tells us, though, is that that the urban environment is causing a breakdown in that sort of natural beneficial interaction between the plants and the microbes. And so urbanization seems to decrease reliance of the plants on the microbiome. So obviously we were scaling, from individuals to communities, and we have a bunch of additional, I'm not going to go into this, we have a bunch of additional stuff that we're working on with respect to habitat fragmentation. What I'm really interested in, and we've been kind of moving towards now, is trying to look at the functional response of the microbiome in this fragmented landscape. And I just want to say that one of the places my lab group is really going is towards looking at those kind of factors within the microbial community themselves. For instance, we've done a study with fire where we've got these 36 different fire legacies of the microbiome over a 50-year fire history, and then cross that with prescribed burns to look at how the interaction between past fire history and future fires plays out in terms of not only the effect on the microbiome, diversity and composition, and their in turn effects on the plants, but most recently we have a paper in review looking at how it affects the response in terms of carbon, nitrogen, and phosphorus cycling. And one of the really interesting things is that atypical fire histories that you would get from fire suppression or fire intensification lead to downregulation of a lot of the genes involved in those functions. Anyway, and yeah, I'm running out of time, so I'm gonna go ahead and say The other thing we've been doing is using a lot of microbial networks to look at community properties within the microbiome. And so we've been showing how stress can destabilize microbial communities and keystone functions within microbes. I'm just going to skip that. The paper's published if you want to look at it. So obviously, I have a real interest in looking at key concepts in ecology through a multi-scale approach, but we've only been able to scale a handful of levels in each of these studies. But I'm hoping in the long term that we're going to be moving from sort of a genes to all the way ecosystems approach. And right now we have a grant proposal in with Jen Rudgers to work at the SEV LTER, where we can look at the functional responses of the microbiome to variability in climate and how that cascades up to impact flux of carbon and so on. So we're getting there very slowly. All right. Thank you, everyone. I'm just going to say thank you to all my wonderful people and my funding sources. Okay. Sorry, guys.
I think we might have time for a couple of questions. I can throw you the cube.
I guess I got carried away. Talked extra.
Ready?
Oh, that's cute.
Really cool talk. I was curious about the mitigation of the oligopathy data that you showed. Did you look at signals for at what phylogenetic scale that was happening at population? Was it the same microbes associated between different plants or different? And if it was same, then have you thought about how those microbes are being dispersed across patches?
Yeah, so good, interesting question. So we haven't looked at the microbiome associated specifically with the plants. We have looked at how the allelochemical impacts the microbial community composition. So the primary allelochemical that they produce is hydrosamnaic acid, which you can buy commercially, it turns out. So we've done experiments where we've applied that and then sequenced the compositional responses. I don't remember I'm sorry to say, I don't remember exactly which groups, but there is a published paper in New Phytologist that one of the things we do in there is characterize which microbes become more common or less common. Yeah. Oh, and we're putting in another grant proposal right now on allelopathy as a mechanism of facilitation for some of those endangered plants. So I'm really interested in that.
And correct me if I got something wrong, but you were saying that there is more beneficial kind of I think you said symbiotrophs in the native kind of block versus the matrix, the urban matrix, but there was also more biomass in the urban matrix. So do you think those pathotrophs are just ineffective? What do you think the relationship is there? Because I assume if there's less symbionts, they would perform worse.
Yeah, so I think the thing is that I don't know for sure, and we've talked about the possibility of doing some manipulations with nutrients, but my guess is that massive nutrient deposition is outweighing the cost of the pathogens, because essentially the plants are just super happy, grow big. So even if they get even if the pathogens are damaging them some, they're still performing well. And so the plants can grow in the urban matrix. Of course, there's also the aspect of impervious surfaces and the fact that people do grow them in their yard as part of some programs, but also I think I think it is less favorable for them, but I don't, in terms of the microbial community, but I just think the microbial community turns out to not matter as much for them in the urban matrix. But I think we would need to be really certain of that, we would need to do some experiments where we actually do field experiments in the urban matrix, which is tricky. But I would like to do that.
Yeah. That's really cool.
Yeah, thanks.
Okay, I think we're all out of time, but maybe we can continue some of these conversations up on the third floor kitchen. But yeah, let's thank Michelle again.